Hello students, today we are going to discuss the complement system. Let us see how does this system works. The complement system, also known as complement cascade, is a part of immune system that enhances or complements the ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism. So, in the previous units, you have already seen how antibodies and phagocytic cells are involved in the elimination and removal of microbes and damaged cells from an organism. So, what these complements actually do, they help antibodies and phagocytic cells by enhancing their role. Okay, they either enhance by complementing it or they enhance by several other means. So, in a way, they help antibodies and phagocytic cells to clear microbes and damaged cells from an organism. It is part of innate immune system which is not adaptable and does not change during an individual lifetime. So, complement system is a part of innate immune system. However, the recruitment of complements is brought about by antibodies which are part of adaptive immune system. Okay, so although they are part of innate immune system, means they are present since birth of an individual, they can be influenced, means their recruitment will be done only by action of antibodies which are generated by adaptive immune system. The complement system consists of a number of small proteins that are synthesized by liver and circulate in blood as inactive precursors. When stimulated by one of several triggers, proteases in the system cleave specific proteins to release cytokines and initiate an amplifying cascade of further cleavage. Okay? The end result of this complement activation or complement fixation cascade is number one, stimulation of phagocytes to clear foreign and damaged material, inflammation to attract additional phagocytes, and activation of cell killing membrane attack complex. Three biochemical pathways activate the complement system are classical complement pathway, alternate complement pathway, and lectin pathway. We are going to discuss these three pathways one by one. Okay? The activation of complement system facilitates pathogen elimination by first one, opsonizing pathogens via surface deposition of complement components 3 C3b. Okay, so opsonization is a molecular mechanism whereby molecules, microbes, or apoptotic cells are chemically modified to have a stronger attraction to cell surface receptor on phagocytes and NK cells, natural killer cells, and thereby they are being removed. Okay, so that is what opsonization means. Promoting chemotaxis of immune cells through production of anaphylotoxin such as complement C5a. Then they are directly damaging pathogen surface through the complement C5b C9 membrane attack complex MAC. We will see all these in detail. So number one, classical pathway. The classical complement pathway is initiated by antigen-antibody complex with the antibody isotype IgG IgM. Okay, so either IgG or antibody IgM is actually involved in the classical pathway initially at the first stage. This activates C1 complex. Okay, as soon as they bind together, the antigen antibody, they activate the C1 complex. Okay, then uh, the C1 complex is composed of one molecule of C1q and two molecule of C1r. So and two molecule of C1s. So altogether it is C1q r square s square. So here you can see one molecule of C1q, then two molecules of R and two molecules of S. So together they are called C1 complex. Following activation, a series of proteins are recruited to generate C3 convertase, that is C4b2b. I will show you in pictures how it works. C3 convertase cleaves the C3 protein. The C3b component of cleaved C3 bind to C3 convertase to generate C5 convertase, that is C4b, 2b and 3b, which cleaves C5 protein. So it is a cascade of events. The cleaved product attracts phagocytes to the site of infection and tags target cell for elimination of phagocytosis. In addition, the C5 convertase initiates the terminal phase of the complement system, leading to assembly of membrane attack complex, MAC. Okay? The membrane attack complex creates a pore on target cell's membrane, inducing cell lysis and death. So this is the picture. Right now we are talking only about classical pathway. So you can see this is antigen antibody complex, okay, and at the FC receptor, FC side of antigen antibody, okay, this C1Q molecule has been bound, okay. Then C1R 2 molecule and C1S 2 molecules, they bind together at the same site. Then after that, what happens is they cleave C4 and C2 into C4b and C2a, and eventually what they do, they with the help of C3 convertase, they get converted into C3. This C3 then again cleaves into C3a and C3b. C3b then goes and combine with other small protein molecules complements to make C5 convertase. Here you can see C4b, C2a and C3b. So this C3b is part coming from above cycle. This C3 after the C3 is broken down into C3a and C3b. This C3b molecule combines with C4b and C2a to make C5 convertase, and then they cleave C5 into C5a and C5b, which in turn, the C5b finally binds with C6, C9, C7, C8 to form MAC, and then they eventually cause lysis of the cell. Okay, and then these molecules C3a in the cycle and C5a, they cause inflammation, and that thereby they bring immunity. Okay, so this is how classical pathway works. Let's see the other one. For alternative pathway, which was discovered after classical complement pathway, the complement factor B, D and P binds to the microbes with lipid carbohydrate complex on their surface. You can see here, that's how they bind to the microbe, okay, with their lipid carbohydrate complex B factor, D factor and P factor. This activates C3, causing it to split into C3A and C3B. So they cleave C3 into C3A and C3B, and then these three, they cause opsonization, cytolysis and degranulation of mast cell. As I discussed with you already, what is opsonization? Again, opsonization is a molecular mechanism whereby molecules, microbes or apoptotic cells are chemically modified to have a stronger attraction to cell surface receptor on phagocytes and natural killer cell. Thereby, they go back for degradation. Then for cytolysis, the osmotic balance of the cell, okay, the osmotic pressure of the cell, it increases, and because of excess water that comes due to salt imbalance, and thereby the cell damages, it gets swelled up and it bursts. Then third one is degranulation. Degranulation is a cellular process that releases antimicrobial, cytotoxic or other molecules from secretory vesicles called granules found inside some cells. It is used by several different cells involved in immune system. Okay, for example, neutrophils, basophils, eosinophils, and mast cells. So that's how they do the degranulation of mast cells. Third one is lectin pathway. Activated by lectin, for example, mannose binding lectin, MBL acts as opsonin and also activate the complement cascade. MBL is made by liver. The lectin pathway is homologous to classical pathway, but with the opsonin mannose binding lectin instead of C1q, this pathway is activated by binding of MBL to mannose residue on pathogen surface, which activates the MBL associated serine proteases, which are MASB, okay, MASP2, very similar to C1R and C1S, which we saw in the classical pathway, respectively, which can then split C4 into C4A and C4B, and C2 into C2A and C2B. C4B and C2B then bind together to form classical C3 convertase as in the classical pathway. Again, this is the diagrammatic representation of the same. You can see all these different pathway, lectin pathway, alternative pathway, eventually they do the same action. Okay, so the classical pathway is activated by C1 complex, then alternative pathway is activated by C3 and C3b, and lectin pathway is activated by lectins, but eventually they go and follow the same route to degrade the microbe and to remove it from the cell. Okay, that's all for today. Tomorrow we are going to discuss cell mediated effector response. All right, okay.